Hello folks, and welcome to a video lecture on meiosis. Meiosis is cell division that starts with a diploid 2N cell that divides twice to produce 4 haploid N cells. This process is necessary as these haploid cells become the gametes or sex cells necessary for sexual reproduction. You see, binary fission in mitosis produces cells that are identical to one another. That is, each cell after mitosis has the exact same copy of chromosome and genes. However, the offspring of organisms that perform sexual reproduction may be similar to their parents and one another. They are each the result of unique combinations of genes. The probability that a second offspring born to the same parents will have the exact same genes as another is 1 chance in 6.27 billion. To achieve this variation in offspring, meiosis performs 2 relatively simple and random tasks. To start, let's take a look at the human life cycle as an example of sexually reproducing organisms. Life cycles of other organisms might be different, but the principles are the same. The gonads of each separate gender produce haploid gametes through the process of meiosis. The male gamete is the sperm, carrying only half the paternal set of chromosomes with a flagellum that acts like an outboard motor, getting the chromosomes to the egg. The egg, or ovum, has half the set of maternal chromosomes, along with all the organelles the first cell will need to start. The joining of the male and female gametes is called fertilization. And fertilization restores the chromosome number to the diploid number. For humans, this is 46. Or 23 homologous pairs of chromosomes. The first new cell is called a zygote that will immediately begin mitosis and develop to form a new individual with completely new sets of genes. Sexual reproduction produces genetically unique offspring because meiosis allows for the reshuffling of genes. 2 events allow the genes to be reshuffled. These events are independent assortment of homologous chromosomes and crossing over of genes between homologous chromosomes. But I'm getting ahead of myself here. Let me explain meiosis first, starting with a diploid cell with unduplicated chromosomes. Let's call the red one the maternal chromosome and the blue one the paternal chromosome. To keep it simple, this example has 1 set of homologous chromosomes. The cell goes through its cycle and copies each chromosome during the S phase. In meiosis I, the homologous chromosomes come together. That's called synapsis. The homologous chromosomes create a double line instead of the single line they did in mitosis. Cell division occurs, but instead of sister chromatids separating, each homologous chromosome goes its own way. This is called meiosis I. I'm leaving out the fact that there are 4 stages each of meiosis I and then meiosis II. Each of meiosis I and II have prophase, metaphase, anaphase, and telophase. The events of these phases are very similar to mitosis, but we'll investigate this detail in class. Each cell is now haploid, but we're not finished here. Another cell division occurs. Each cell produced in meiosis will divide again, and this time it divides more like it did in mitosis, in that each sister chromatid separates into individual cells. This process is called meiosis II. Each cell has 1 copy of the chromosome, so it's still haploid. Independent assortment is a random event that can take place in meiosis I. Let's go through the process again here quickly. This time, we'll need to use 2 sets of homologous chromosomes. Again, the red are maternal and the blue are paternal. The chromosomes just happen to line up like this, with the paternal on one side and the maternal on the other. Meiosis I finishes and the homologous pairs are separated. However, there's a chance that the homologous pairs could have lined up this way, with the maternal and paternal alternated. When meiosis occurs, each cell can get an alternate copy of the maternal and paternal chromosomes. For each possibility, there are different outcomes after meiosis II occurs. with just 2 sets of homologous chromosomes. There are 4 different chromosome combinations in the gametes. This can be mathematically calculated using the formula here, 2 to the n power, where n is equal to the number of homologous pairs. In this case, n is equal to 2, so 2 to the 2nd power is 4. 4 different possibilities. Humans have 23 pairs of chromosomes. So 2 to the 23rd power is equal to 8,388,608 combinations of chromosomes in the production of gametes. With just independent assortment, there's a 1 in over 8 million chance that 2 gametes will have the same set of chromosome combinations. Those are low odds. But independent assortment isn't the only thing occurring that mixes up the outcome. Something else happens during Prophase I of meiosis. Remember that during Meiosis I, and particularly Prophase I of meiosis, homologous chromosomes come together in a process called synapsis. The combination is now called a tetrad, which refers to the 4 sister chromatids. Remember, only homologous chromosomes do synapsis, so one here is a maternal and the other is a paternal chromosome. Homologous chromosomes carry similar genes for the same traits. For this example, we'll use traits you're familiar with, eye color and hair color. While each sister chromatid carries identical genes, its homologue carries a variation of that gene. So let's say the paternal chromosome has copies of genes for brown eyes, while the maternal chromosome has copies of genes for blue eyes. Now, when synapsis occurs, another random event takes place. The 2 homologous chromosomes will appear to embrace one another and exchange segments of their sister chromatids. These segments contain genes, so really, it's the genes that are getting exchanged. This event is called crossing over. The sites where crossing over occurs are called chiasmata. Now, we have 2 new chromosomes, each with new gene combinations. Again, in this simplified version of meiosis I and II, with the result of crossing over, 2 divisions occur, resulting in 4 completely different gametes. What's interesting to think about is that the crossing over event is completely random. In each instance that it occurs, sometimes only a small number of genes are exchanged, while in others, large numbers are exchanged. The number of genes exchanged is a random event. After meiosis, another chance event can occur that increases the likelihood that offspring have different combinations of genes, and that is random fertilization. That's when the roughly 250 million genetically different sperm try to fertilize the randomly ovulated egg. To summarize, 2 events in meiosis created genetic variation: independent assortment and crossing over. Random fertilization adds another random event to the mix. Jerry Hirsch, a geneticist at the University of Illinois, calculated the probability that any 2 human parents will produce 2 offspring by separate fertilization events with the same combinations of genes will be equal to 1 in 6.27 billion. Can you think of why this variation in the population of any species is important? Write down your response and prepare to discuss this in class. Well, that's it for meiosis. I hope that was helpful and that you took some good notes. If you have any questions, please write them down and we'll see you back in class. Until then, be well.
For me, nature is one of the neatest things on this planet. That's why Rodney and I started Nature Walk, because we want everyone to know how neat nature is. How neat is that? That's pretty neat.